Okay, now let's talk about the energy needs of the cell. The only type of energy that the cell can use is ATP, which stands for adenosine triphosphate. It consists of adenine, which is a nitrogen base that's also in DNA and RNA, ribose sugar, that's the same sugar that's in RNA, and then 3 phosphate groups attached to it. If we remove the last phosphate and break that bond, we release energy. And then we're going to add the phosphate back, rebond it, and store energy. So it's going to be a constant cycle that's happening in our cells. Now, there's 3 ways to make ATP. The first way is substrate level phosphorylation. And this is where we're going to take this phosphorylated compound that contains a phosphate, and we're going to take the phosphate away from that and transfer it to ADP, adenosine diphosphate. Diphosphate has 2 phosphates, and we're going to add this other phosphate from this phosphorylated substrate to it so that we have 3 phosphates and we've made ATP. The second way to make ATP is oxidative phosphorylation. And this is just a series of redox reactions that happen during our respiratory pathway. Then there's photophosphorylation. This happens only in photosynthetic organisms. Here we're looking at what ATP looks like. And you see here's the adenine, the nitrogenous base, the ribose sugar, and you have 1, 2, 3 phosphates. That is ATP. When we break this bond where the energy is and release that phosphate, we have released the energy in the bond, and we have made ADP, adenosine diphosphate. And if we're doing the substrate level phosphorylation, we can add that phosphate back to it from a phosphorylated compound, and we can make ATP again. Now, without the second or the third phosphate, we just have AMP, adenosine monophosphate. The reason the cells need energy is because most of the things the cells do require energy, and so it has to be ATP. Cells need it for respiration, for fermentation, for photosynthesis, and they use it for chemical work, for transport work, and mechanical work. Now, the pathways of bioenergetics, is mainly we're going to use glucose. It is the primary catabolism of glucose, and it goes through 3 coupled pathways: glycolysis, the Krebs cycle, and the respiratory chain. The processing of nutrients varies, but basically these 3 processes convert glucose to carbon dioxide and give off energy. The first one and the most effective is aerobic respiration. It involves a process called glycolysis, the Krebs cycle, and the respiratory chain. And in this, oxygen is the final acceptor of electrons, and so we have the capacity to make up to 38 ATPs through aerobic respiration. Anaerobic respiration doesn't have oxygen as the final acceptor, and we're not going to make nearly as much energy. It varies as far as how much ATP we make depending on what the microbe is, but it involves glycolysis, the Krebs cycle, and the respiratory chain. Fermentation also happens without oxygen being the final acceptor. Instead, you're going to have organic compounds as the final acceptor. And we're going to be able to make a maximum of 2 ATP molecules, so it's not very effective. Now, I put this slide in here to summarize what we call glycolysis. We use 1 glucose, and we split it into 2 pyruvates. And we partially oxidize the glucose or the sugar, and we reduce 2 NADH molecules. We use 2 ATPs. We make 4, so that gives us a net production of 2 ATPs. And the end product is those 2 ATPs and 2 pyruvic acids. That NADH is what we call an electron carrier, because when you split molecules like glucose, they give off electrons, and these electron carriers, NADH, pick them up and carry them, and when they pick them up, they become NADH, and they cash them out at the end.